Hello, everyone, and thank you for tuning in to Help for HDTV. Today, we have Dr. Ed Wild, who needs no introduction. Everyone in our community knows and loves Ed Wild. And we are so thankful for him to come on today and answer some questions that the community had submitted. So welcome with us, Ed. We're so excited to have you.
Thank you so much for having me. It's great to be here, and I'm always happy to ask questions as long as I don't mess up my scheduling too badly.
It's all good. Our first question we have is, I have an adult child at risk for HD. I believe we should all participate in research to advance knowledge. As the healthy parent, I would like to find out what my two healthy alleles are. Other caregivers have been talking about this. It's easy now for us to get this information because of at-home testing, places like HD Genetics. What do you think? Would knowing both parents' CAG be helpful for research in the future?
That's a really interesting question. So the thing about having two healthy alleles is that people who are in that situation can be really helpful to research studies, but their role would be as control participants. So when we're studying Huntington's disease, in humans, we study people who have an expanded copy of the gene, or which we call an expanded allele. And those people are the kind of Huntington's disease people that we learn about the disease from. But we only know what's happening in the disease by comparing them with people who don't have expanded alleles. Broadly speaking, when we do Huntington's disease research, we get a group of HD expansion carriers and a group of controls, and we do something like brain scans or thinking tests, and then we find out what's altered in the disease. And then the CAG count, the length of the expanded copy of the gene, comes into play really when we're looking, if we find a difference, we then look at the length of the CAG in the group with HD to figure out whether the CAG is making a difference to that particular thing. So for instance, in my research, we looked at a protein called neurofilament in the spinal fluid and blood. And we got a big group of controls and a big group of HD expansion carriers. And we found that the neurofilament level was much higher in the body fluids of people with the HD expansion, which tells us that we can use this protein to measure how much damage is happening to neurons. Once we had established that the level was higher, we then looked at the relationship between the CAG count and the level of protein. So if you got 10 people whose CAG was 42, whose age was 42, but then some of them had a CAG of 60 and some of them had a CAG of 39 or something, did the people who have a big CAG have a bigger neurofilament? And the answer was emphatically yes. So the CAG count's really important for research when it comes to figuring out, once we find a disease-related change, what's the relationship between the gene and the change that we have found? And can that help us to either give people useful information about their lives and their bodies, or could it be something that we could use for getting people into trials more efficiently and running the trials and testing the drugs? The interesting thing about the size of the gene in people who don't have an expanded copy, so healthy controls, is that it's almost always between 15 and 20. Very rarely is it outside that range. And in that range, we don't see a great deal of impact on any of the biology that we've looked at in humans. So as people get older, for instance, if we just think about the neurofilament protein, as people get older, the neurofilament protein just tends to rise very slowly because, as we know, as we get older, our brains slowly shrinking. Sorry about that. But it's true of everyone, whether we have HD or not. It happens faster in people with HD, but everyone's brain is slowly shrinking. And as far as we can tell, on things like that, the CAG in the Huntington gene doesn't make a big difference. The times where it's been shown to make a difference is Marcy McDonald, who's one of the most brilliant in HD, took some cells in a culture in a Petri dish and looked at the efficiency with which the cells were using energy. And in the normal range of CAG, it turns out that if your CAG is a little bit higher, your cells are actually more efficient at using energy, which is a bit of a clue as to why the CAG tends to expand. I will get to the answer in the end, don't worry. I just give long answers. It's a clue as to why the CAG has tended to expand as humans have evolved. And the answer is that basically a little bit more CAG probably makes your cells more efficient and therefore allows your brain to be the incredible computer that it is. And the Huntington's disease, in the words of Nancy Wexler, Huntington's disease is too much of a good thing. So the CAG has tried to expand, and almost always, up to a point, that's been a beneficial thing for the development of the nervous system. But then there's this tipping point around 37, where if you go over that range, the protein starts to become toxic and you start to cause damage. So the question was, can healthy people who are from the unaffected side of the family, can they get their CAG tested? I think the answer is yes. I don't know how you could do it, but that's probably just because I'm working in the UK and our healthcare system is paid for by the government. It's the National Health Service. And therefore, genetic testing is really done on the basis of risk. And so it would be the only times we do Huntington's disease testing is if someone is known to be at risk of HD because of an affected parent, or if they've started to develop signs or symptoms suggestive of HD, which presumably isn't the case here. The other context in which it can happen is in the research setting. If you're a volunteer in the Enroll HD study, it's likely that if you enrolled as a control participant, it's likely that your CAG has been measured in a lab, but that's done in a completely anonymized way, and those results are never fed back to control participants. So what you could pay to have it done privately. What's the advantage of it? For you personally, I don't think that there's any knowledge really that comes from knowing what your own CAG lengths are. I'd be quite curious to know what mine are, but it's not something that I would spend money on finding out, because whether it's probably going to be 15 and 16 or 15 and 19 or something like that. And then it'd just be like, oh, okay. It's not useful information, and it's probably not going to tell you much about yourself. Could it help with research? Yes, but if it's going to be helpful for research, it'll be something that's folded into the research anyway. Sometimes we just don't need to know the CAG, and for control participants, we just wouldn't test it because HD is so rare in the general population that the chance that a control participant will suddenly turn out to have a big CAG is very low. So to avoid the risk of accidental disclosure of something that someone might not want to know about, we just don't test it, or we test it in that kind of anonymized fashion where it's completely separated from the participant and the care team versus the lab team. Could it be something? Oh, yeah. So what I was going to say is there is a potential downside, though, which is you could go into this kind of sending your DNA away to have it sequenced and thinking that the answer would be interesting. Wouldn't it feel great if I had 2 CAGs of 19? Because maybe that means my cells are more efficient. But actually, something like between 0.5% and 1% would have one allele that's expanded a little bit into the kind of intermediate range, which we call the gray area. And that comes with a bunch of kind of doubt and uncertainty and not much useful information. So if I did this and I discovered that one of my CAGs was, say, 30, what that means for me is I'm not going to get Huntington's disease from that. But it's possible that if I had a kid, that kid might then inherit an expanded allele. And then that's not information that I was looking for when I embarked on this journey of curiosity. But then that becomes a big thing for me to think about. But actually, if I say to a genetic counselor, I've got this CAG of 30 or 31, what does this mean for my kids? The answer is going to be those alleles are so rare and they haven't been well studied. And it's so different from one person to another. We actually can't give you any useful information. All we can give you is a bunch of kind of unknowns. And so I think the reason it's not offered routinely, and the reason why people, even in research, are not control participants are not given that answer, is basically because of the answer. The answer can be stressful, but it's never going to be useful. So there's no upside really.
So it's so funny you say that, because I didn't submit this question. I'm just asking it. But of course, I follow your research. All of us do. And so I literally wrote at the bottom of my paper, what about your research with the general population if they found out they were in the gray zone? Because I know about your research of testing a healthy population. And so when you test a healthy population, what is the percentage that you find of people that are actually in the gray zone?
It's about 1%, somewhere between 0.5% and 1%. But there's the kind of light gray zone and the dark gray zone, if you like. And so the lower gray zone numbers are closer to 1%. The higher gray zone ones are rarer than that. But it's not been that well studied in the population. And it's interesting because actually a lot of the genetic research we have about the frequency of things in the population that are genetic comes from big genome studies. But actually, until very recently, the technology that's used in those studies is pretty good for sequencing nearly everything except CAG repeats, because the way that the gene frequencies are counted is basically by sticking kind of measurement DNA molecules onto the DNA from the person, and they stick on like Lego bricks. But if you've got lots of CAGs in a row, which everyone has in the Huntington gene, the ingredients that are there to count the number can get lost and can get stuck or lose their way in the middle of that big CAG repeat. So that's why when we do a Huntington's disease genetic test, we have to do this very specific test that's either putting a marker on one side and then the other and measuring the length of DNA in between, or we use a special, much more expensive method to actually add on the letters one by one, which is less likely to get lost in the repeat. And that's a higher level technique that actually probably isn't available. I may be wrong here and I'd have to check, but something like 23andMe, I think even if you wanted it to do a Huntington's disease genetic test, I think the technology used would actually struggle to perform a Huntington's disease genetic test to the required level of accuracy that we need. But so that's why we don't really know the exact answer to the frequency of the gray zone expansions in the community, because community DNA studies have largely been based on technologies that don't measure those CAGs particularly accurately. But certainly through people, the 50% of people who don't inherit an HD mutation but do a predictive test, in those people, obviously the CAG is measured accurately, and that's where we get that kind of roughly 1% figure from.
Yeah, and I wonder if this question comes more from like onset to people are thinking, because it's so hard to predict the onset. And we just did an FDA meeting with her, and it was public knowledge. So I can say because she's put it out to the public. Both her and her husband, fiance, are both at risk for HD. And they ended up having a child, CAG expansion of 115, diagnosed symptomatic at 2, and she's at end stage of life at 7. Dad isn't even symptomatic yet. Either is. Right. And so they're both not symptomatic at this time. But they've been said, oh, this probably came off the dad, but neither one of them has tested. But she was wondering if I'm in the gray zone because my grandmother got it so late. Would that play a role?
Okay. Oh, so maybe I slightly misunderstood. Everything I said is true and very interesting, but may not be an answer to the question. So let's try again. So what I would say in that situation is that where there's a very big expansion from one generation to the next, it almost always comes from dad. And that's because men are constantly copying DNA in order to make sperm, whereas women making eggs, the DNA isn't being copied all the time. And so that's why almost all cases of juvenile or pediatric Huntington's disease, the allele did come down the line from dad. As to sort of each person obviously needs to make their own decision about whether they would like to get tested. There is a lot of information that's encoded in the big allele and not much information that's encoded in the little allele. And really the only way to answer that question would be to do a predictive HD test and go through the allele size individually and discuss what it can and can't tell us. Increasingly, as there are people who are slightly perplexing in that we think, how have you got to that age without having, we know you've got a repeat size of whatever, how have you got to that age without having Huntington's disease yet? And two people with the same CAG, one of them could have onset in their 30s and the other might be in their 90s. And it's been a source of great frustration, but we are actually making big strides into figuring out what those differences come down to. And in a lot of cases, it's either little changes within the CAG repeat from a CAG to a CAG, which can be tucked away inside the repeat, or it can be little single letter changes in other genes, genetic modifiers. And a lot of this work has come out in the past 6 or 7 years from studies in the Enroll-HD DNA database, genome-wide association studies coming from the GEM-HD consortium. We're starting to understand what those mean. And I think like at some point in the future, when someone opts to have a Huntington's disease test, I think we may end up saying the test was positive and you have this number of repeats, but it's good news because you have a CA interruption here and that predicts slightly later onset, plus you have a favorable mutation here in this other DNA repair gene. which statistically is associated with slightly earlier onset. And it's not like we can then say it'll be Tuesday, the 5th of March, 2061 that you start to develop symptoms. But we can hopefully start to get a little bit better than where we are now, which is this is your CAG number, and it doesn't tell us much apart from the fact that you'll get Huntington's at some point.
Thank you so much, Dr. Wild. And I'm so glad you gave Marcy a shout out. I think everyone knows Dr. Wexler, and she's so amazing in the whole group, but Marcy McDonald's rarely talked about. And I had the privilege of meeting her a couple of times, and she is an amazing researcher, and we wouldn't be where we were if it was not for Marcy. So
she's one of my favorite people in the world, honestly. She's incredible.
Yeah.
And if she's watching this, she'll be blushing.
She's true. She is
very modest, inappropriately modest.
Yes. So our next question is, if Prilenia trial is approved, what are the next steps to getting the approval drug to the patients? We hear that it takes a long time for families to get access after approval. Why is that? Is it something because of labeling or regulatory? Is there pathways we can help as a community to make the process faster?
So the Prilenia trial of predopidine, which is called Proof-HD, is one of the phase 3 trials. And the idea there is that if you basically, this is a shot on goal, right? And it's all pre-agreed with the FDA. You set your endpoints, you set your sample size numbers. If the drug meets its endpoints, you get your primary endpoints, you get a big green tick, and it's essentially guaranteed that the FDA and the other regulatory agencies will grant a license for that drug in the indication in which it was tested. Why does that take a while? That's complex, and it's not just regulators, but it's also payers. And I'm not an expert on the US healthcare industry, as I say, but if a drug really knocks it out of the park, and if this is known to be a disease with extremely high unmet need, such as Huntington's disease, if there's no doubt about the drug meeting its endpoints and demonstrating itself to be safe, then actually these things can happen really quickly. For instance, nusinersen, which is the first drug that ever worked for spinal muscular atrophy. I'm going to pull figures out of my brain from 2015 here. I think that the announcement was made that the drug had been successful sometime in September of that year, and the FDA issued a license for the drug on Christmas Eve of that year. So that is quick, right? I think that was the quickest approval the FDA had ever issued. But the point there was, this was the first drug in a neurodegenerative disease with higher levels of unmet need, and the drug completely aced it on all of the endpoints. So there was no question. And why did it happen so fast? Partly because it was obviously a good result, partly because the families of the kids affected by SMA lobbied the FDA, and partly because I think the FDA is a bit better than it used to be at recognizing when decisions need to be taken quickly. So then what's the holdup? Some things happen more slowly than that. Usually it comes down to the data being less clear. So it might meet its primary endpoint, but not make the secondary endpoint, or it might be that there's a sign of a signal in some patients, but not others. And if it misses the primary endpoint, then maybe on some combination of secondary endpoints, you can get a kind of conditional approval, but all of those things will take a lot longer to negotiate. And then there's the question of who's going to pay for the drug. And in the UK, it's the government, and it's the government bodies that decide. In the US, it's the insurance companies, but the decision-making process is the same, which is basically the company sets a price, and the people who are paying have to decide whether the price is worth it. And what this comes down to is a calculation about how much it costs to not give people the drug versus how much it costs to give people the drug. In other words, they look at the life of someone with Huntington's disease if it's untreated, and that's an expensive thing for insurance companies to pay for, or the state to pay for, in terms of care, symptomatic treatments, and lost tax-paying income. And it sounds brutal, but this is how these calculations are carried out. Excuse me. On the other side, though, is how much you spend on the drug and how much difference that would make to the quality of life of the person, and by extension, the cost to the insurance company of keeping that person alive. And this is a big negotiation. So the example I gave in nusinersen, in Europe, the drug was licensed by the EMA, the European Medicines Agency, almost as quickly as the FDA, but then it took 3 years for that drug to be prescribable by doctors, because all of the healthcare agencies of Europe basically said the company's asking for too much money. The amount that they're charging for the drug makes it financially unviable to prescribe the drug. And eventually the company said, okay, I can't make money by selling no drug, so we'll make it cheaper. And once they did that, then the calculation changed and everyone was able to start prescribing the drug, and it finally was able to start being given. But that was 3 years in which children died
and suffer
from a disease that was actually known to be treatable. And it was everybody's fault. The company asked for too much money and the government held their line, and everybody knew that would cause delay, but nobody wanted to be the ones to concede, because they both had a lot of skin in the game. So these things can get messy. I think that, and actually this is somewhere where families can make a big difference. I think the first thing to do is to look at the data, okay? And if the data look good, if the trial meets its endpoints, then everyone will be pulling in the same direction. And the job of families is to make that happen as quickly as possible by lobbying their insurance companies and lobbying the regulatory agencies. If the data don't look good, I think or if it's borderline, I think it's important to accept that it's what it is. And we shouldn't be lobbying the agencies to license a drug that didn't work or that didn't meet its endpoints. So I think we have to start with the data, and then if the data look good, that's when it's really important for everyone to have a kind of focus on getting these discussions happening as quickly as possible.
Sure. Thank you for that. And then I'm gonna push it over to Josh, but I can't imagine like having a child suffering with SMA and waiting 3 years on the sideline, knowing other people in other countries are getting it. And I know that it's greed and money, but for all of us that have children at risk for Huntington's and stuff, I would go insane knowing there was something there for my kids and I could not get access to it. But that's a whole nother story. Go ahead, Josh.
I completely agree. I can't say anything other than that. I completely agree. But these agencies are large, and their job is to be immovable. The job of the governments is to have a certain amount of money and to spend it as efficiently as possible on improving. And the same for the insurance companies. Obviously, they're trying to make a profit as well, but their job is to basically spend the money they have as efficiently as possible. The job of the drug companies is to make their money back and then make a profit. And so you've got these competing objectives, and everyone knows that every day that those decisions are not taken, children and people with these diseases are suffering. But their answer is, we're doing this as quickly as we can, and if only the other people compromised, we'd be etc. It was very frustrating. We got there in the end, but it was super frustrating.